Effects of osmosis in living organisms. The cells, tissues, organs, and systems of living organisms are always exposed to body fluids or solutions of different concentrations. This is true. There are 3 types of solutions an organism may be exposed to. We have hypotonic solution, isotonic solution, and hypertonic solution. A hypotonic solution is one whose concentration is lower than the concentration inside a living cell. This is hypotonic. A solution whose concentration is lower than the concentration inside the living cell. Then isotonic solutions have no net osmotic effects in living organisms because a dynamic equilibrium exists between them, okay, and the living cells. So nothing will happen. Hypertonic solution, on the other hand, is one whose concentration is higher than the concentration inside a living cell. Okay? Now, let's look at this. Let's look at the effects of osmosis in animals. When an animal cell, such as a red blood cell is placed in hypotonic solution. Hypotonic solution meaning a solution which has less concentration than the solution inside the tissue, inside the red blood cell in this situation. Remember, the water molecules will move from the region of from the less concentrated region to the concentrated to the more concentrated region. So what will happen here when you place an animal cell like a red blood cell in hypotonic solution? Will it lose or gain water? Okay. Now, hypotonic solution has less concentration than the concentration inside the living cell. So the red blood cell will gain water by osmosis. Why? Because the water potential of hypotonic solution, the water potential, okay? The water potential of hypotonic solution is higher the water potential inside the cell. In other words, the concentration of hypotonic solution is lower than the concentration inside the cell. As a result, it swells and eventually bursts. The bursting of an animal cell due to osmotic gain of water is called cell lysis. Are we together? It's called what? Cell lysis. When an animal cell is placed in a hypertonic solution, a concentration which has a higher concentration, an animal cell will lose water by osmosis. Why? Because the water potential inside the cell is higher than the water potential of the hypertonic solution. Or if you want to say in terms of concentration, the concentration inside the cell is less than the concentration of the hypertonic solution. As a result, it shrinks and wrinkles or crinkles. The shrinking and crinkling of an animal cell due to osmotic loss of water is called crenation. Okay? Nice. It's called crenation. Osmotic loss of water by animal tissues leads to dehydration. That's the effect. It leads to dehydration of the animal. This is an illustration. Yes, a good illustration. This is a normal red blood cell. Okay? A normal red blood cell, if it is placed in hypotonic solution, it gains water and eventually bursts. Cellolysis, or it is cellulized. Okay? If it is placed in hypertonic solution, it loses water. The process called what? Crenation. Okay? So, therefore, it is not advisable to take a lot of water, because there will be what? Cellulitis, which is a problem to the body. So don't always, there are a lot of people who say drink a lot of water, water is important. Yes, water is important, but anything in excess is bad. Okay? Anything in excess is what? Is bad. On the other hand, again, if you just drink too much alcohol, for instance, what's that brand? First Choice. Okay? First Choice. What will happen is that you will lose water. The cells will lose water. Okay? And you'll be dehydrated. The process is called crenation. Okay? Crenation. Now, that is the effect of osmosis in animals. Now, let's look at the effects of osmosis in plants. Okay? Effects of osmosis in what? In plants. Now, when a plant cell is placed in a hypotonic solution, like in animal cells, it gains water by osmosis. Okay? The reason is just the same, because water potential of the hypotonic solution is higher than the water potential inside the cell. But the result, what is the condition, is different. The condition is what? Exactly. As a result, its protoplasm swells and eventually starts pressing against the wall. In animal cells, it bursts. Here, it starts what? Pressing against the wall, the protoplasm. Now, what do you call the condition where the protoplasm of a plant cell presses against the cell wall due to osmotic gain of water? Correct. It's called turgidity, not cellulitis. In plants, it's called what? Turgidity. Are we together? A plant cell that is undergoing turgidity is said to be turgid. Now, why doesn't the plant cell not burst? Because it has a cellulose cell wall, which is what? Which is rigid, which is strong. Okay? Now, what happens when a plant cell is placed in hypertonic solution? A solution whose concentration is higher than the concentration inside the living cells. What will happen is that it, like in animal cells, it loses water by osmosis. Okay? The reason is just the same. The water potential, because water potential inside the cell is higher than the water potential of the hypertonic solution. Remember, if you have high water potential, then in terms of concentration, you have lower what? Lower concentration. If you have higher water potential, if you have lower water potential, you have higher concentration. It's opposite. Okay? Now, in animal cells, when a cell loses water, that is called crenation. Okay? It shrinks and wrinkles. But in plant cells, its protoplasm shrinks and pulls away from the cell wall. This is called... Now, what do you call a condition where the protoplasm of a plant cell pulls away from the cell wall due to osmotic loss of water? I'm waiting. Correct. Plasmolysis. Okay? So a plant cell that has undergone plasmolysis is said to be plasmolysed. Okay? Said to be plasmolysed. We have an illustration here. This is the normal plant cell. Now, when it is placed in hypotonic solution, the protoplasm process increases and the presses against the what? Against the cell wall, like this. Okay? Then when it is placed in hypertonic solution, plasmolysis takes place. The protoplasm it loses water, and this causes the protoplasm to pull away from what? From the cell wall. Okay? So if we say, what is the name of this party here? This party. Okay, just let me do this. This party here, it is the cell wall. Okay, that just moved because of it has been what? Plasmolysed. Are we together? Very good. Very good. Now, listen to this information. When a plant tissue, such as a peeled potato tuber, is placed in hypotonic solution, it gains water by osmosis and becomes bigger and more firm. The presence of water in plant tissues forms a hydrostatic skeleton, which renders mechanical support to the entire plant. Okay? When a plant tissue such as a peeled potato tuber is placed in hypertonic solution, it loses water by osmosis and becomes what? Flaccid or flabby, or small and weaker. In a living plant, this leads to a condition known as wilting. Wilting is sagging of delicate plant parts such as leaves, flowers, and the young stems due to what? Due to loss of what? Water. Temporary wilting is one which can be reversed by supplying the plant with water. Okay? Now, permanent wilting can not be reversed even if a plant is supplied with water. The plant tissues have already died. Okay? That's why it is not... Now, you should also know why it is not advisable to apply too much fertilizer on plants. Okay? Why shouldn't you apply too much fertilizer on plants? Okay? Think of in terms of the concentration of fertilizer and the concentration inside the cells in plants. Okay, I'm waiting for that one. Just write in the comment section. Okay? Otherwise, your work is coming. Now, let's look at the final. Let's look at the last thing, and transport across the cell membrane. Active transport. The movement of particles against a concentration gradient using energy from ATP is called what? Active transport. It is the main process by which mineral ions move into or out of the cells. For instance, ion uptake by root hairs and uptake of glucose by epithelial cells of the villi is an example of these are examples of substances that that move by active transport. Okay, uptake of minerals and also glucose is by active what? Active transport. Okay, water molecules, osmosis. All right. Then dye, like crystals, colored crystals, that is diffusion. Okay. Okay, so we've looked at cell organization, transport across the cell membrane, that is diffusion, osmosis and active transport. Now you can answer any examination question as long as it's coming from cells, transport across the cell membrane. Okay, as long as it's coming from there, you can answer any question. Are we together? Very good.